Seeds, embryo, endosperm. So this is a diagram showing parts of a dicot seed. A dicot seed has got two cotyledons, and in the middle what you see is the embryonal axis. So it comprises of plumule, which becomes a shoot later, and then radicle. So this is the diagram showing monocot seed parts. A monocot seed has got a seed coat, fruit wall, alburon layer, scutellum, which is the cotyledon, and the embryo. The embryo has got plumule and radicle. Plumule is protected by coleoptile and radicle is protected by coleorhiza, and monocot seeds have got endosperm. Seed coat is also present in dicot seed, though it is not labeled over here. Now this is a diagram showing the parts of embryo in a dicot seed. The first picture shows the textbook one, showing the two cotyledons, and in between you can see the plumule. The plumule, and then below the plumule comes the epicotyle tissue, which will form the leaf primordia, and then comes below the level of attachment of cotyledon comes the hypocotyle tissue, which will become the future stem. Hypocotyle terminates in radicle, and a radicle is having a root cap. The same we can see in the next picture, where a dicot seed is germinating and they have labeled hypocotyle there. Hypocotyle will become the stem of the plant. It terminates in radicle, and then above the level of attachment of cotyledon, what tissue is present, that is called epicotyle tissue, which will develop to form the leaves and the shoot. So you can find in the second picture two cotyledons. So hypocotyle comes below the level of attachment of cotyledon, and epicotyle is a tissue which arises above the level of attachment of cotyledon. Monocot embryo parts. The first picture shows maize. We can see the endosperm clearly in maize, and the second picture shows the structure where the cotyledon is placed towards the side. It is called a scutellum. Then we can see the plumule as well as a radicle, supported by structures like coleoptile and coleorhiza respectively. Now some of the seeds have got endosperm in mature seeds. They are called as albuminous seeds. For example, castor, you can see in this picture, endosperm is present and two cotyledons and the embryo which is present in the center. Other examples of albuminous seeds are coconut, maize, wheat and barley. So this is the coconut endosperm. The first one in glass is a free nuclear endosperm of coconut, and what we see inside the coconut is this white part is the cellular part of endosperm. And you can see how the embryo derives nourishment. So this is the embryo of coconut. The white structure which is found in the center is its cotyledon, and the cotyledon will derive its nourishment from the surrounding endosperm. You can see the embryo, leafy embryo coming outside the coconut. Endosperm in maize. Examples of non-endosperm or non-albuminous seeds are pea, beans and groundnut. So this is a picture of groundnut. We can see the embryo clearly arising from one of the cotyledons. The two cotyledons can be seen, but we cannot see any endosperm because it got used up in the mature seeds. The other example of non-endospermous or non-albuminous seed is peas. Peas scientifically called as Pisum sativum. You can see the cotyledon, you can see plumule, radicle, hypocotyle tissue, seed coat, but you won't find endosperm in the mature seeds of pea plants.